This is Rochelle, your Microbiology IT tutor. Welcome to today's Microbiology retention session where we will be studying DNA replication mechanism. Now, if you need study resources to help you get that A, then grab your Microbiology IT workbook. The link is in the description box. DNA replication mechanism. So our first branch here is semi-conservative model. From there, we have the daughter DNA molecule, which has one parental strand and one new strand. Okay, too many loops. So that's semi-conservative model generally. So each daughter DNA molecule has one strand, one parental, and one new strand. Our next branch, we are going to tackle the strands. And from here, we have two branching out. We have the lagging strand and leading strand. With the lagging, you will have to remember that it's synthesized discontinually. And we have Okazaki fragments. And many primers. So there's many primers on the lagging strand. And then Okazaki fragments are the nucleotides placed on the strand. And then eventually the primers are lifted up where more nucleotides can fit in, and then the ligase will come and seal everything up. So we have many primers. And onto the leading strand, it is synthesized continually. Or continuously, actually. And it's synthesized 5' to 3'. Make that 5 a little bit better. 5' to 3'. And you see here on the notes, replication fork dynamics, that the leading strand, the one that we just completed, is synthesized continually towards the fork. The lagging strand is synthesized discontinuously away from the fork in Okazaki fragments, later joined by the, like I said, DNA ligase. Our next branch is enzymes. Now, we have a ton of enzymes here that participate. So let's go. We have helicase, and a key word is unwind. We have SSB proteins. They're single-stranded binding proteins, and they are used to protect and prevent to protect from degradation and prevent reannealing and prevention. And our next enzyme is primase. Key phrase here is RNA primers. They lay down the RNA primers. Next enzyme is DNA polymerase 3. And you'll need to remember extends the primers. Next, we have DNA polymerase 1. Removes and replaces. So the DNA polymerase 1 removes the RNA primers and replaces DNA. So removes RNA primers and replaces DNA with DNA. Two more enzymes. We have ligase, and ligase seals the nicks. And then we have DNA gyrase or topoisomerase, and it prevents overwinding. Prevents overwinding. Winding. Okay. So those are our major enzymes that are associated with the process of DNA replication. Our last branch is going to be split into two. Proofreading and repair. Proofreading, we have DNA polymerase 3 that is involved in proofreading. It's also involved, well, the whole process is the 3' to 5' exonuclease activity, which is the removal of nucleotides that are mismatched. So this is an important thing to remember. 3' to 5' exonuclease activity. Okay. And then we have repair, and that involves DNA polymerase 1. And that is the primer removal. And repair. Okay, so now we have our micro mind map. Quick recall fill-in. Number 1, blank relieves supercoiling ahead of the replication fork. Again, you can put your answers in the chat. Use your micro mind map to answer these questions. Number 2, short RNA segments laid down to initiate replication are called number 3, the fragments formed on the lagging strand are called. So take advantage of your micro mind map, take a look, and use it to help you answer these questions. And then you can put your answers in the chat. Okay, number 1 for the multiple choice. Which DNA polymerase is the primary enzyme of replication in E. coli? The micro mind map is your friend. So use it, take advantage. It helps you to quickly jot down major topics and then allows you to expound as you explain it. And then it helps you to answer these questions. Number 2, the enzyme responsible for sealing nicks in DNA by forming phosphodiester bonds is. Remember, when you're answering these questions, pick out the key words. So with number 2, the enzyme responsible for sealing nicks. Okay, sealing nicks sounds familiar. Let me go back to my mind map. Where have I seen sealing nicks? And then you'll be able to find your answer to number 2. Number 3, proofreading during DNA replication is primarily carried out by, again, what's the main word? Proofreading. Okay, where have I seen proofreading on the mind map? Go to the mind map, find proofreading, and that will take you to your answer. Complete the sequence of replication steps at the fork. So helicase unwinds DNA, and then blank prevents reannealing. Then primase lays primers. Then DNA polymerase 3 extends the strands. Then DNA polymerase 1 removes the primers. And then blank seals the gaps. So you're responsible for filling in those blanks. And then you can put your answers in the chat. Okay. Application case. So a bacterial mutant lacks functional DNA ligase. Predict the effect on application on replication of the lagging strand and explain why the leading strand is less affected. So when you are looking at these application cases, make sure that you are focusing on the major points. So 1, here we have a bacterial mutant lacks functional DNA ligase. Okay, DNA ligase. What does it do in DNA replication? Remember that. Then it says, predict the effect on replication of the lagging strand. Okay, what is the lagging strand? I remember it has something to do with Okazaki fragments. It's synthesized discontinually, and it has many primers. Okay, so now, and then it says, and explain why the leading strand is less affected. Okay, what is a leading strand? Well, it's synthesized continually, 5' to 3'. So there aren't any Okazaki fragments. It's very smooth. So take all of that information into effect, and then you're able to come up with the answer to this application case. I'll see you in the next interactive tutoring session. Let's microbiology it.